Welcome to the Salk Institute's Where Cures Begin podcast, where scientists talk about breakthrough discoveries with your hosts Ali Akmal and Brittany Fair.
I'm here today with Salk staff scientist Gerald Pao. He studies a myriad of topics, including the novel coronavirus and how to make animals transparent. Yes, you heard me right, transparent. So welcome to Where Cures Begin.
Oh, thank you for having me here.
And thank you for being on the podcast today. And just to start out, are you originally from San Diego?
No, I came to San Diego when I was 17 years old, but before that I grew up between Spain, Germany, and the East Coast a little bit. My dad was a career diplomat, and he decided to put us in German school since I was in kindergarten. I was in the German schooling system until I finished my high school graduation, but at times I was in New York. There was a German school in New York, and then I was also in boarding school in Germany. So I've been around.
Wow. And what was it like hopping between these three different places that have three different cultures and three different languages?
Well, I speak all four languages. So I speak Spanish, German, English, obviously. And then I speak, both of my parents is Chinese. One is, my dad speaks Mandarin, my mom speaks Cantonese. It was okay. It was not confusing at all. People assume it's to be confusing, but it's actually easy if you're learning all these languages within your critical period.
I see. And when did you first become interested in science?
Oh, well, I became interested in science probably late in kindergarten, but definitely before first grade. Initially was with an encyclopedia and then encyclopedia of animals, and then I just ditched all the encyclopedia parts that were not science. So there was a clear interest like really early on. When I was in fifth grade, I learned about DNA replication. So I saw a documentary and understood how DNA replication would work. But then I went to my biology teacher, who was a phage virologist, and then I asked him how gene expression in a genetic code worked. And he said, well, you'll learn it when you grow up. That was obviously not satisfying. So I went to my dad, and then he sent me the Center of Molecular Biology to do science when I was in fifth grade, or maybe sixth grade.
Oh, wow. So you mean you were actually going to a university?
Well, I was not going to class in the university, but I was in a lab in university under a postdoc from Caltech since the time I was 11 or 12.
And what was it like to be 11 years old in a lab of adults?
Well, it's like I felt I was behind. So I was trying really hard to catch up. He gave me a bunch of books to read. One was biochemistry, the other one molecular biology, and then those were the sort of like the main ones that I started with at that age. The professor was much more of a mentor. He's the one responsible for me ending up in the Salk. He's the one who told me to come to UCSD, and because the UCSD is also associated with Salk, and that's where you should go.
And why was UCSD and Salk the place you should go?
Because at the time, it's like I told him I wanted to do molecular biology. I thought Princeton was probably the best one I got in, so I even paid the dorms in Princeton. Then he told me, what happened to your application at UCSD? And I says, well, I never heard back from them. And then it's like, then called the registrar, actually was admitted. They just forgot to send me the notification. He says, oh, you should definitely go to UCSD because that's where a guy called Dan Lindsley was at UCSD, and also the Salk, and there's also where Francis Crick was. So he told me to go there.
That's true. Nobel Prize winner Francis Crick was here. Did you ever have a chance of meeting Francis Crick?
Yeah, but just as the undergrad that, is on the side of the conversation. But I would run into him because he had the next door office of my boss at the time, yeah, I would see him frequently, putting up posters on the floor and then making cutouts of different parts of papers and then sticking them, gluing them on to make a poster to organize his thoughts. That was interesting.
Oh, wow, that's really interesting. And you've also mentioned to me before that you had a very different type of job when you were in high school. Can you tell me about that?
Well, yeah, so I was going to this gym, and then a booker in like Madrid's oldest modeling agency saw me there and then kind of asked me whether I wanted to work. So he put me to do stunts because I was doing gymnastics, to do stunts for commercials, and then went into modeling fashion stuff. So pretty much I saved up money to go to college from modeling in high school.
That's incredibly impressive. Did you model for any big brands or anything our listeners might have known?
First, as like being Asian, you did not have many opportunities back then in Europe. And but there was one brand that was celebrating diversity, and that was like Benetton. And then I did every season of their runway for about 3 years.
Oh, wow. So you were actually on the runway modeling fashion.
Yeah, that was in Madrid, Barcelona, Milan, Paris. I mean, it was some kind of thing that you could actually make substantial amount of money without interfering too much with high school.
And you did decide to come to UCSD for your undergraduate degree, but it sounds like you had opportunities to come to Salk at times. Is that correct?
Yeah, it's like one of the professors at UCSD sort of formally introduced me to Tony Hunter, actually sent me to his office. And then because to work on some signal transduction, computational stuff that I was doing back then.
And what was your first impression of Professor Tony Hunter?
Encyclopedic knowledge of cancer. He seemed to be the one who knew the most. Anyway, I still think that nowadays.
Did he have a beard back then as well?
Yes. I have never seen Tony without a beard.
Well, I have to ask, was it intimidating to interact with people like Francis Crick and Tony Hunter, who were really leaders in their fields?
Yeah, it's totally intimidating. Around there were all these cartoons, scientifically themed jokes about oncogenes and signal transduction. I remember because I was so nervous, I wanted to be there super early. I was there almost an hour early and just looking at the stuff and waiting until he came.
Okay. And today, you're now a staff scientist here at Salk. Can you just explain to our listeners what it means to be a staff scientist? Because people may not be familiar with that title.
A staff scientist is basically a semi-independent position under a professor at Salk. It's basically you're more senior than the postdocs, and you can apply for your own grants. And then basically you're working more or less independently at that point, while receiving advice from, the professor.
So you're a staff scientist in the lab of Tony Hunter, but you're also pursuing numerous different independent projects. So to start out, I would love to hear about your project involving Squid.
I had a friend from the climbing gym. Her name is Gerald Pao. And then she told me about these proteins that existed in squid, and they have really high optical refractive index. So refractive index is basically the speed at which light will go through a particular material. What that does is it bends light. So for example, if you actually look in a swimming pool, you can see the light gets bent. For example, if somebody's standing there and you look at them from the side, sometimes you see like these really, really short legs on somebody. It's an optical illusion because the light gets bent and then it makes it look like the legs are shorter. So this protein actually is super dense. It's much more dense even than glass. So this is very, very, very strange. So my idea is basically like, okay, if you can make it so that the refractive index of things is the same, between two different materials, then you can make it transparent. So what do I mean by this? So the problem that we're dealing with organisms is a little bit similar to what you have in a cloud. In the cloud, you cannot see through because the light scatters, although the cloud is made of water and air, right? So, but the water itself is transparent and air itself is transparent. But the fact is that you actually have these tiny droplets of water, and every time that the light goes through between air and water, it bends. So it actually gets bounced around many, many times through the cloud because there are many, many droplets of water. And then you cannot see through. You just see white because the light gets scattered. So just imagine that if you were to take the space in between the drops of water in the cloud and fill it with something that has the same density of water. So just imagine it's water for simplicity. So if you fill all the spaces between the droplets of water with water, then it would look like a giant floating swimming pool of water in the air. You could still see through it, but it would be transparent. So we're trying to do the same thing in tissue of the organisms. So the tissue is basically, you have fats that are high refractive index, similar to the level of glass. And you have the in-between space, the cytoplasm, extracellular space, and inside the nucleus. Those are pretty much filled with water. So we're filling that space in between with this reflectant protein, diluting the reflectant protein in to match the same refractive index of the fat membranes. And this way, in theory, you should be able to make things transparent.
So you're trying to make tissues transparent. I mean, that sounds super cool, but why would you want to do this?
So there are techniques that use this principle to make tissue transparent, but it involves killing the organism. But if you were to see, let's say, something that I look at, for example, brain activity, obviously you cannot do that with a dead mouse. So if you were to have to see, other than the surface of the brain looking for activity, then you would actually have to make it transparent somehow. So that's sort of the major reason, is like we wanted to look at brain activity while the mouse is living. So that is sort of the goal.
Okay, and I have to ask, because I just watched X-Men, could this technology be applied to humans to make someone transparent?
Right now, it's still not efficient enough to make a human like that. But in theory, you should be able to. The fact is that you do have transparent organisms in the animal kingdom. There's a deep sea fish, many people may have seen, that the front of the head is transparent and the eyes of the fish actually look through its own head. So these things actually do exist. So transparency is actually compatible with life. It's just that it didn't happen in mammals. There was no selective pressure to do this. So I think that maybe, with genetic engineering and some cleverness using some things that nature evolved, we might be able to get to that point.
That's absolutely mind-blowing, but crazy cool. And if you're studying these squid proteins, I mean, how do you get a squid protein in the first place?
The protein itself we actually make in the lab, but the genes that were required for this, we actually have to sample in the wild. We started a collaboration with University of Tokyo and our Riken Brain Science Institute in Tokyo, in Japan, to get these squid. They're called big fin reef squid, Sepioteuthis lestoniana. So when you actually start doing field work, the most critical thing is to work out your logistics. You have to find collaborators, you have to find ways to ship your samples back, you have to find, and everything has to work smoothly. So especially with squid, because the squid you see in the market or the supermarket and buy it and eat it, that stuff is not in the quality that you require for cloning genes. Actually, if you don't freeze this thing immediately within 5 minutes, then you don't get any RNA that you can clone. So in order for you to get the RNA sequence, that you need to actually freeze it immediately, flash frozen. So I was going together with a fisherman, and then they actually bring the squid alive, and then at the dock, basically take the sample from the squid and then freeze it with dry ice and ethanol. When you are doing field work, you have to rely on the weather. For example, you can only sample certain times of the year. So that's either in July or in November. Both of those seasons, especially the July, it's typhoon season also. So basically you have to you have to allot at least 14 days or so, hoping that you get one day where you're lucky, where you can get adequate samples.
And then you also have some interesting studies involving essentially a fake version of the coronavirus. Obviously, we now have a vaccine, but there's still so many things we have to learn about how the coronavirus spreads and what it sticks to, among many other things. Can you tell me about that project?
We're trying to make things that are mainly useful. So what do we want to do, and what can we do? One is that we found that we make viral-like pseudotype viruses and viral-like particles. So pseudotype viruses are basically viruses that are based on HIV, a lentiviral vector. So a vector is a virus that has a single round infection. It cannot reproduce. It just goes into the cell. But in this case, you make the cell, you carry this green fluorescent protein and makes the cell green, so you can actually see it. And then we coat it with the protein from the SARS-CoV-2 coronavirus. And this allows us to test the properties of the SARS-CoV-2 spike protein. And this spike protein is basically how it enters into cells. So then it's like we can actually test which mutations are better, which mutations are worse than entering into cells. And for example, we found that this mutation that happened late in the Chinese epidemic that made its way to Europe, this called spike D614G, this actually infects 5 to 8 times better than the original coronavirus. So it's much more infectious. So pretty much everywhere where this mutant showed up, it took over. So we demonstrated in the lab that this actually has a basis, and that is in the lab that the virus actually did something that actually makes it more infectious, and we showed that. So in addition to that, what we did is basically we incorporated this green fluorescent protein. So now basically what you have is viral particles that are actually fluorescent. So you can actually see them. So you could actually use these and see, okay, how do these spread in aerosol? People assume that every aerosol particle, for example, will actually have viruses, but nobody has actually put viruses in it. How many can you fit inside? Can you actually do have certain, does this actually, to which surfaces does it stick, for example? So you can actually put these viral-like particles, because we can visualize them. We can see, does it stick to a face mask? This face mask of this material versus the cotton one versus the surgical one versus the nylon one that you have, on which one does it stick? Does it go through? Does it go to the other side? And then you can actually measure all these kind of things. You can actually measure whether the stuff gets stuck in the air conditioning filter. What kind of washes take the virus off? You can test all these things because you can then go and see if the virus is there by fluorescence, and because it glows green. So we made these, and we're just testing these right now.
That sounds extremely useful. And I know a lot of people are worried now because we're indoors and it's winter, that circulating air, especially through the air conditioning, could be spreading the coronavirus. Is that something you could possibly easily study with these fluorescent proteins?
We can actually monitor both the droplets and the droplets that have viral particles, and how much of them and how many of them and where they go. So,
okay.
So that's in the works.
And you have all of these different projects going on, lots of collaborations. What is your favorite part about being a scientist?
Well, my favorite part of being a scientist is to do things that have been underexplored. I go for the things that are considered basically high risk, high reward. And they're risky. They're risky because so little is known, or nothing is known, that it's very likely that whatever you guess was wrong. But if it has potential, then it's maybe worth taking this risk.
And just kind of a fun question here. If you were no longer a scientist, what would you do? Did you ever consider another career, a backup option?
Nope. Yeah, I've never considered, I probably would not want to live if I was not a scientist.
Oh, wow.
So yeah, I don't think I have any interest in living if I was not doing science in some capacity.
And what do you think the future holds for your field?
So for me, personally, I don't think I am in a particular field. Because, for example, I just got accepted a paper in computer science. I do like systems neuroscience. I do theoretical neuroscience. I did molecular biology, virology, kind of material science with the squid stuff, a little bit of squid biology because of necessity. But I think I'm not really tied to any particular field. But that's the great thing about being staff scientist, at least for me, that I've been able to be truly develop skills to be interdisciplinary.
That's a really good point. So let me ask you a different question then. What do you see as the future of your work?
So the future for my work, so we were able to do this kind of brain download finding. And this is basically what we're able to do is like we could actually take calcium imaging from brains and then convert them into a computer model that reproduces everything that in this case was a fly, that behaves like a fly and walks like a fly based on the neural data. It's sort of the general concept of like downloading a brain, but in reality, it's not just science fiction. So now what we're doing is like we want to make it into a new type of AI that's based on real biology. Right now, the AI that exists is basically purely computational. It's sort of generally vaguely inspired in biology. But this one is basically really starting from real observations of real organisms and let it learn in the computer at the speed of computers to do new things. So we want to make a new type of AI that's completely based on biology.
That's insane. So if you're able to put the brain of a fly, essentially upload it to a computer, and then have that computer start acting like a fly, is your goal to eventually do that with other animals? I mean, could a long-term future be that humans could upload their brains to a computer?
Yeah, I think so. I think we've at least the mathematical basis to actually do that. I think we have a pretty good grip on that now. Yeah, so I think that might be possible, but you might be limited to whatever you record. So for example, if you're recording yourself walking, you can only reproduce yourself walking, maybe a little bit more, but you might have to have recordings of you doing the variety of things that you normally do in order to be able to download yourself completely.
Well, I'm so curious to see where your work goes, and it was fascinating learning about all of your different projects. So thank you so much for joining us on the podcast today. It was a pleasure speaking.
Yeah, thank you for having me.
Join us next time for more cutting-edge Salk science. At Salk, world-renowned scientists work together to explore big, bold ideas, from cancer to Alzheimer's, aging to climate change. Where Cures Begin is a production of the Salk Institute's Office of Communications. To learn more about the research discussed today, visit salk.edu/podcast.